until we pass it on to Abe, is Axel Huntelmann, who is a guest researcher in the Institute for the History of Medicine at Charité University in Berlin. His research deals with the modern and medieval history and political sciences, as well as business administration. How do you manage to put those together? He has worked and published in the on the German imperial hospital system and other European and public health institutions between 1850 and 1950. The history of laboratory animals, a biography, and this is my favorite part on the immunologist Paul Ehrlich, who a remarkable, I mean, he'll tell us all about him. The book is superb, by the way. Paul Ehrlich, of course, in one fell swoop of just rational thinking, started the field of immunology by saying antibodies are produced by selection, not by instruction. And they had to fight a couple of very important people to do that. So, Axel, it's a pleasure to have you. The title of the talk, Interconnecting Centers and Margins, Technologies and Other Infrastructures in Bacteriology and Immunology Research, 1870 to 1930. Sir.
Thank you very much for the kind introduction and for inviting me here. I think it will be difficult to get all the things, centers, margin, technologies, networks, immunology into one paper. We will see if this works well. And I'm thankful to Miguel because it fits so great. We just moved 100 years earlier. And the technique as such is not that sophisticated as Miguel. So this picture represents a network of global actors in the context of the Iran-Contra affair in the 1980s. So this network is a result of long-standing investigations of Mark Lombardi. Beside the fact that it is also a piece of art which could have been visited in the Whitby Museum in New York for modern American art, it's also a network, and that gets straight ahead to our conference or to our workshop. Who and where is the center here? And who and where are the margins? This is a question. I will come to this later on. It becomes even more difficult if we enlarge the network. So, and to find here margins and centers will be probably another difficulty. Only in a focused way, you might be able to differentiate different centers and margins. For instance, and I will come back, this might probably be Paul Ehrlich, this might be Robert Koch or somebody else. In this case, this is the network of Oliver North and so on. And a very important thing here is I argue that networks are only snapshots because they are constantly shifting, changing, mutating, and so on and so forth. And this is exactly because you can see here, this is a network 4th version. So he just added new context and so on and so forth. So decades ago, the sociologist Mark Granovetter has differentiated between strong and weak ties in human relationships and argued in his groundbreaking article, The Strength of Weak Ties, that a network of broad numbers of weak ties is more sustainable and more flexible than strong ties. So I would go further in this aspect, especially I'm dealing with the question of innovation and knowledge transfer, and on a very small snapshot, so to say. Granovetter's argument, I argue, has some consequences for the topic of the workshop and for my paper. First, interconnections between centers and margins are mainly weak ties because it's so time-consuming to entertain strong ties. And a network of many weak ties is very volatile, and the idea of a stable network is an illusion. And a network of numerous weak ties demands an intense social or relational economy to balance all the different interests of all the people involved. I call this relational economy, so it's some sort of a moral economy. And a network of weak ties is, as I said, constantly mutating and changing. What I will do in the following minutes, something 30, 35 minutes, My starting point is the ceremonial opening of the Staten Serum Institute in Copenhagen. And I will come back to this group later on all the time. And from there on, I will go back to the 1870s and show in what way the network has completely different and how this network has evolved. And then I will go into the main innovation at that, not the main innovation, it's a very practical innovation, but on this innovation, sidechain theory based upon, it's a very basic evaluation of serum, the efficacy of serum, and how there are actor networks involved, and how these distributional network of standard serum played a major role. We go into network pedigrees, kinship and patronage systems, and Ehrlich's relations to Britain, United States, and Eastern Europe. And in the end, I will conclude. So the Staten Serum Institute in Copenhagen had been inaugurated in September 1902. For the official ceremony, life scientists from Denmark, Britain, Germany, Italy, and the United States traveled to Copenhagen. Several Danish ministers attended the festivities, and the accompanying program included official speeches, an 8-course gala dinner, a guided tour through new institutions, and to sightseeing tour through Copenhagen. And among these visitors were Julius Morgenroth from the Royal Prussian Institute for Experimental Therapy in Frankfurt, the Institute's director Paul Ehrlich, George Dean, head of the antitoxin department at the General Institute for Preventive Medicine, and others. At the gala dinner, Paul Ehrlich was placed prominently at the head of the table between Carl Julius Salomonsen, professor of bacteriology at that time and director of the new institute. On the other side of Ehrlich was sitting the Prime Minister of Denmark, Johan Henrik Deunsner. Despite the character of an official ceremony, the inauguration showed also elements of a private family celebration. Ehrlich and Deunsner were framed by the married couple, Salomonsen. Mrs. Salomonsen, on the other side, was sitting next to Wilhelm Hermann Olof Madsen, the father of Thorvald Madsen. He later on became president of the hygiene organization, and Salomonsen's scientific assistant and member of the new institute, and he was a de facto director, and in 1909 he became, so to say, real director, official director of the Staten Serum Institute. At the Grand Serum Party, so it was called, during the inauguration ceremony, visitors of the celebration were photographed in the private rooms of Thorvald Madsen. So he was living also in the institute. I'll show you a photograph later on. Overall, the photograph presents on a first level scientists and visitors in the context of the inauguration ceremony. As so many conferences, this photograph is about spinning the web. Some of the represented scientists barely know each other, or some get to know each other on this occasion. For instance, Paul Ehrlich is here, number 7, get to know with Svante Arrhenius, at that time already a great scientist. A year later, he got the Nobel Prize for chemistry. And they first cooperated, and then they hate each other. But that's a different story. On the contrast to these loose connections, some of them are colleagues or having a close working relationship, and some are even family members. The ties could be followed by reading the Festschrift that had been published on occasion of the opening. This is a Festschrift title and Salomonsen's introductory words. And the interesting thing from the Festschrift here, the first sentence, and we are talking about globalization, so this is about globalization. The Festschrift starts, we have a Festschrift in the Danish Institute, written in English, and it started in the autumn of 1894, after Roos' publication of the first decisive experiment with Behring's therapy applied to diphtheria. The production of anti-diphtheric serum was eagerly taken in hand everywhere. So, I mean, this is international. We have Roux from the Paris Institut Pasteur, we have Behring from Marburg, and this altogether. But on page 6, he goes further. And I just read this, and I will come back to the photograph and to this quotation all the time at the end. I only state that our method of immunization follows very closely up on that applied by Dr. Dean at the General Institute for Preventive Medicine. It is Dr. Dean, too, whom we have to thank for so kindly supplying us with a race of diphtheria bacillus, Park and Williams number 8, it was a special strain, which we are now using. The methods of measuring are, of course, those of Ehrlich. This eminent pathologist has, during the whole existence of the department, assisted in every possible way, and I take the opportunity to thank him. This is more or less a quotation about talking about networks and the positive effects of networks and so on. So on a second level, we could reconstruct some of the individual networks, for example, Salomonsen's weak ties, and differentiate closer working relationships he held to the Pasteur Institute, British Institute of Preventive Medicine, New York City Board of Health, where he distributed the Park and Williams strain, which had a very high and constant toxigenicity. At the center, and I'm coming back to the photograph, at the center we find without doubt Ehrlich. He's centrally seated at the gala dinner, prominently mentioned in the Festschrift, and he is located in the center of the photograph. He is here, number 7. Arranged in the way of family photographs, Ehrlich is not only located in the center, but also framed and surrounded by family. So look at, here is the father of Thorvald Madsen, Olof Madsen. He was a war ministry in Denmark. This is his wife. Here is Thorvald Madsen, his sisters. This is the wife of Paul Ehrlich, sitting with them, family. This is Carl Julius Salomonsen and his wife. So we have this family structure within this whole network. Beside these family ties, there are other strong genealogical ties. Scientists stood in the tradition of a certain research field, and they defined themselves as scholars of somebody else. For instance, Thorvald Madsen defined himself as a disciple of Salomonsen and of Ehrlich. Because he spent there quite a couple of months in Frankfurt. As a whole, members of an institution, a research institute, but also as a thought community, could become a close community as described in Sandra Legout as a famille pasteurienne. So you have this very close, I mean, it's an institution, but it's seen as we are family. To sum up, the quotation in the photograph illustrates a scientific network with weak ties between different scientists and strong ties between family members, colleagues, hierarchical relations between directors and their employees, and also genealogical relations. At this snapshot during the official inauguration of the Staten Serum Institute, it seems to be clear that Ehrlich was at the center. Definitely, Salomonsen and the Madsen family played an important role. But who was at the margin? So, for instance, Johann Fibiger. I mean, he is really a margin here. He got the Nobel Prize in 1926, and he is a disciple, a doctoral student of Salomonsen. He got a professorship in 1900, so he is clearly here at the margin, but he's not really at the margin. Or take, this is a number, Karl O. Jansen, who first developed a transmittable mouse tumor by inoculation, and whose research became essential for Ehrlich's Institute, and also for the Cancer Research Fund in London or in Harvard for William Council. So he is number 15. You can't really see him very well. Yeah, there is on the, but he's also on the margin here. But in fact, he's not. He becomes very important for the different scientists. So this is a question: what is the margin? What is the center? It seems to be that the General Institute in London is also at center in Salomonsen, who was designated as the founding father of bacteriology in Denmark. It's much more a center for the Danish scientists than Paul Ehrlich. So it depends on the perspective. And it becomes even more volatile over time, as we will see when we go back in time. I will come back later on to this. You see also here the different names. German since Woodhead, I will come back to him later on. And Karl Weigert, just to call him here, he is a cousin of Paul Ehrlich, the older cousin, and he is also a good pale of Salomonsen. Yeah. And William Ballack is also a very eminent figure of bacteriology. So they were all here. Going back in time, Ehrlich and Salomons met first in summer 1877, just 25 years ago. Salomons already had finished his medical studies. He was working as a hospital physician, and he was somehow settled. He finished his dissertation in 1877 and decided then to go to Wrocław to gain some further knowledge on breeding bacteria and to work together with Julius Cohnheim and Ferdinand Kohn. Salomon at that time had already published a very well-known article on the isolation and breeding of bacteria, and in his dissertation, Salomon had developed a method to conserve blood and to cultivate microbes in small glass tubes. So then he went to Wrocław in 1877, and Cohnheim's assistant, Karl Weigert, remember, he was here at number 10. Oops. This is Karl Weigert again, at the margin, so to say. He became a good friend of Julius Salomonsen, together with William Welch, who was at the same time in Wrocław at that time. Weigert's much younger cousin, Paul Ehrlich, was still a medical student in summer 1877, but even if Salomonsen and Welch met Ehrlich regularly at Cohnheim's Institute, the young student participated only very rare in their leisure activities, as Salomonsen remembered decades later. Another outsider of the group was the completely unknown country physician Robert Koch, who visited Wrocław from time to time to go into the library, work there, and have some discussions. So looking backward, and here you see this of summer 1877, here you see Salomonsen. I think it looked a little bit like a Berlin hipster cafe with all the people with long beards and all the stuff. So and here, Karl Weigert, and this is William Welch in his younger years, and he was also finished his medical students, and after he has worked, he went to Wrocław. So looking backward, it becomes clear that center and margin have shifted. Salomonsen and Weigert were much more closer to each other and to the center, whereas Ehrlich and Koch were completely at the margin. The network relationship between Koch, Ehrlich, and Weigert and Salomonsen changed over the next 20 years. In 1878, Ehrlich went to Berlin to the Charité Hospital, as well as Koch, who moved 2 years later and became medical counsel at the Imperial Health Office. Whereas Weigert moved to Frankfurt, to the margin of science. So he was Since the early 1880s, Ehrlich and Koch had a close relationship, and Ehrlich described himself as a disciple of Koch. Protected by his official status, Koch became the main proponent of germ theory of disease, which was very controversially discussed, but as a medical counselor of the Imperial Health Office, he had the support of the director. After he identified the tuberculosis and cholera pathogens, bacteriology and scientific hygiene became the leading discipline in science. We just had this already. For the identification and visualization of germs, a set of devices, special technical knowledge and experiences of laboratory processes were necessary to cultivate, preserve, and visualize specific pathogens. The technical processes of visualization became more important than the pathogen itself, as Thomas has illustrated and a decade or 2 decades ago. So I don't go into this anymore because this is completely well done. The new bacteriological methods and techniques were now taught in special courses at the Imperial Health Office and later on in Koch's own institute. So medical officers, military surgeons, and physicians from Germany all over the world traveled to Berlin, having some training, working in the institute. So here you see the 2 students. This was the best way, 2 students, but overall, this is one course of students at the Kaiser Wilhelm Academy, so at the military, the Academy for Military Surgeons, which made courses. And this is one of my favorite pictures because Koch in 1890, he really looked completely annoyed and exhausted, And this is Emil Behring, who really hated teaching. And this is really, you can see how he was. So whatever. So this is, I mean, you get an idea about the number of medical officers, military surgeons, physicians, and so on, went to get trained, went to Robert Koch and get trained. And among them, Carl Julius Salomonsen, who visited Koch several times. So they discussed open questions in bacteriology and met on conferences. For instance, here in 1902, this is Paul Ehrlich, Robert Koch, and this is supposed to be Salomonsen, according to a photograph I found in the Ehrlich papers. So this is in 1901. During his visits in the 1880s and early 1890s, Salomonsen also met Ehrlich, especially after Ehrlich became a member of Koch's Institute in 1891. Their cooperation intensified as Emil Behring, Ehrlich, and others at Koch's Institute of Infectious Diseases were working on the development of the diphtheria antitoxin in Berlin, as well as Salomonsen did in Copenhagen. Especially Torvald Madsen, who was working on this topic and on the constitution of the diphtheria toxin. Before and after the breakthrough of Behring and Ehrlich in Berlin and Drew in Paris, the biggest problem was the industrial upscaling of the antitoxin, respectively serum, to produce serum with a stable antitoxin titer and to evaluate the serum's efficacy, just to compare them on the market or scientific progress in the work on the improvement of the serum. In this context, Ehrlich played a major role, and again, the network relationship changed, because until then, Ehrlich was a hospital physician. His main mentor, Frerichs, he died in 1884. He was completely lost, so to say, and he started from 1891 at Koch's Institute. This is one of the, to understand the problems of evaluation. So what you can see here, this is the process of intoxication of serum horses, and this you can see here is the toxin. The animals that get intoxicated develop as an immune reaction antitoxins, and later on, after a certain titer had been reached, there was a bloodletting, coagulation, and then that's it. The serum was sold on the market. And this is why it was so important to get the Parker and Williams strain number 8, because it depended on the strength and the toxigenicity of the bacteria strain to develop a much higher antitoxin titer, which could be sold for more money and could be more effective to the patients. So this is more or less the overall process, but you have a biological. You don't have a chemical substance. You could just measure or weigh or something like this. And that was the problem with the evaluation process. So after diphtheria antitoxin had been marketed by private serum producers in Germany since 1894, serum distribution was regulated by the state. Every serum produced for the German market, every serum batch for the German market had to be tested on purity and its efficacy, approved by a state-run institute, and the new director of the institute became Paul Ehrlich. In 1897, he developed a new method of evaluating the serum's efficacy, which was a complex relational system, including a standard or so-called normal serum, standard toxin, and the serum sent in by the producer. The reference base was a vacuum-dried standard serum. So these are different files. I mean, this stored in a closed glass file. This was one of them. You could just broke it up, and then you can use it. And here's some sort of the powder, just to give you an idea. Every 2 months, a glass tube was broken, diluted in water, and used as a base to calibrate the standard toxin. It was that they were, so to say, eternal, have the same efficacy, and they won't change over time. But the bacteria, the toxin, changed over time, and so it was necessary to recalibrate them every 2 months. And this recalibration and the estimated efficacy was expressed in immunization units, which was just a coincidental unit. For the approval of diphtheria antitoxin, the serum sample was mixed with a quantity of standard toxin that corresponded to the amount efficacy of the produced serum. I'll just go further on, then it can. In the sample's efficacy, if the sample efficacy was exactly as announced by the producer, the guinea pig died at the end of the 4th day. It was exactly after the 4th day. If the guinea pig was still alive on the 5th day, that was great, because the serum was much stronger than announced, so that was great. But if the guinea pig died earlier, the serum was rejected, sent back to the serum producer, and they just had to trash the whole serum batch and to start from the very beginning. So it was a very expensive process for the serum producer and had very strong consequences. So I will not go deeper and skip further details, but I think what becomes apparent is that serum evaluation was a complex process and even more complicated with other sera. Whereas for the evaluation of diphtheria antitoxin, only 2 guinea pigs were used. There was 2 medical officers, 1 for each, 1 control animal and 1 mouse for the preservative to check if there was any intoxication. For the evaluation of tetanus serum, already 2 strains with 8 mice, exclusive all the control animals were used, and you can count here. And for the evaluation of erysipelas serum, 154 mice were used. So various factors influence the evaluation process and the relational system, which is an splendid example for an actor network if one want to do this, because you have all the different factors just to mention. You have the bacteria with the culture medium temperature, you have the serum, you have the scientists which have to get trained, you have the serum producer and the horse as a serum dispenser, you have the test animals, and you have the recipient, the patient. Probably he is not suffering only from diphtheria, but also from tuberculosis or other diseases. And if the serum didn't work, that probably does not mean that the serum did not work, but because he has other infectious diseases. And also the devices and instruments. And the aim was synchronization, standardization, and stabilization of this process, which was very complicated. For Ehrlich, it was not only necessary to stabilize the actor network en miniature, but also on a broader and social level. He formulated detailed instructions about the practical process for the serum producers. He published his research results explaining the sidechain theory, which was behind. And the interesting thing is, it's similar to you. There was first the practical processes, and from the practical processes, he started to explain how it works and develop, so to say, or develop further the sidechain theory. And finally, serum producers and scientists from all over the world could visit his institute, where the process were explained personally and practically. In these years, Salomonsen visited Ehrlich more than 1 time, and Madsen did an internship for several months at Ehrlich's institute. In an article, he compared the British, French, and German system of evaluation and concluded that due to the extraordinary importance of their therapy, one has to say, though, probably I have not gone into this, but therapy is, if you will find nowadays the therapeutic for cancer, it's the same as xerotherapy at that time. I mean, it was the first effective drug against the cause of the disease. So the importance of serum therapy, and I quote Madsen, the serum's potency should be evaluated based on an international scale and unit, because it is possible only in such a way to compare and to appreciate correctly the successes obtained. And as such an international scale, he proposed the German method of evaluation. Because, as he said, the method was reliable and easy accessible. What started as an explanatory correspondence, occasional demonstrations, or training to colleagues who visited the Institute for Experimental Therapy soon became institutionalized. Guest researchers visited the Institute for a training. I will just go into this. This is only why this is so important. So it's a quantification and objectification of an efficacy that could not be clearly measured. And now I'm coming just to the practical training of the work instructions. And the standard serum was shipped to all other serum producers. And there was a transmission of technical knowledge and know-how by official guidelines. beyond this, there were national scientific interests. And there is a quotation in one of the guidelines that the normal serum and the institute was a master of measurement. And the normal serum was compared with the Urmeter in Paris, so to say, as a standard. And in 1913, 56 institutions worldwide get standard serum and took the German system as a, and there was mainly, except France, most of the institutions who produced serum. So coming back to the training. And after these young students went back to their home institutions, they started to establish the German method at their home, asking for standard serum, and then it was sent to their institution. Soon a subscription system was established and standard serum dispatched every 2 months to serum producing and regulating institutions. And within 10 years, the German system became as a reference magnitude. So, as I said, the institutionalizations. First, we have some training with friends like Salomons and so on and so forth. Then we have courses. And one of the first institutions receiving standard serum as a subscription system is Salomons, #1. Then we can see here George Dean, Theobald Smith from Boston. I come back to Pawlowski in a couple of minutes, and Arthur, who is also on the photograph. Arthur is here, number 27, who is here. So he also, they were all on the list. They received standard serum from the very beginning. Besides bacteriologists, we already know from Grant serum therapy, also Alexander Dmitry Pawlowski from Kiev, and Kozlov from Rostov, and Stefan Korshun from Charkov were among the first who received standard serum. Their connections to Ehrlich followed similar patterns as already described. Ehrlich met Pawlowski, for instance, on a conference in Paris. On his way back, he made a stopover in Frankfurt. He got trained in the institute. He went back home, and then he was writing a letter to Ehrlich. Said, please send me standard serum. I want to establish the system here. And even more, they were corresponding all over Russia, and other institutions sent in a letter to Ehrlich. Could we do some training at your institute? Could we get some standard serum? And the subscription system was expanded. So overall, from the 56 institutions all over the world, half of the institutions were in Eastern Europe. What becomes more apparent is here when I'm talking about subscription systems and organized courses and training, the institutionalization. Here you can see very soon you could make training courses which have to cost money, but you get a working desk, probably one, for instance, Korshun, who had to pay laboratory animals extra. They had to pay a fee to stay there to make the laboratory training. One effect was they had an additional income. And here you can see the lists like Shiga, who was very famous from Japan, later on working on chemotherapeutics with Ehrlich together, Möb and Kies, I come to him back later, and here Konstantin Levaditi from the Pasteur Institute. And this is what you can see here. He got a short letter by Eli Mechnikov recommending him, as many other letters, to work at the institute. So this is more or less this, if we're talking about pedigrees and about who was working with who, this is clearly, you can clearly go back into this. This is from Ascoli. I'm probably going further into this detail. And also from these, so to say, if we have a network, this institutional or family network, you can see this is a photograph of the members of the institute and guest researchers. And we'll just go just to show the relationship between the researchers there and Paul Ehrlich and others. For instance, this is Shiga there on that side. Nogard is a guest researcher, and this is Preston Kies. He was a disciple of William Welsh, coming to the institute, working with Hectoan in Chicago at that time. And in 1904, he organized a visit for Paul Ehrlich in the United States, and he was celebrated, so to say, as a hero for whatever, getting an extra professorship in Chicago, and so on. And just to get into this, and if we are talking about a famille and a disciple and father and son relationship, this is a letter he wrote to Ehrlich in 1906. Please take the best care of yourself, and with it the grandson often. So this was a complaint of Ehrlich that he had at the time. I cannot begin to tell how much I miss you. My father died 17 years ago, and in all that time since, I have had no ideal companionship of an older man except with you. Indeed, no father could have done more for me. Oops. Than you have done for me, and my gratitude is far beyond that. So this is, so to say, a strong tie relationship into the United States, and this is why he organized all these different visits for Ehrlich and so on and so forth. And he stayed every year for a couple of months in Berlin. When we're talking about all these corporations and institutions, this is Paul Ehrlich on one of his daily tours through the Georg Speyerhaus, a research institute. And here we have, and this is a different aspect, I'm coming back in a minute. Here we have the cooperation of different scientists. And this is Tashiro Hata, one of the cooperators that led to the development of Salvazar. Okay, this is more or less the idea of working relationships within the institute. So if we assume that the Institute for Experimental Therapy and Ehrlich is a center, and the margins depended on the supply of standard serum for information, or as I will show, on chemical preparations, we have to consider in opposition that the center depended on the margins, their cooperation and their need for standard serum. If the margin did not accept the center, its authority, the superiority of interpretation, and did not verify the scientific results, or if they even ignore the scientific work, the center would not be the center. And furthermore, the so-called margins like, one might say, Copenhagen, New York, or Charkow were centers from a different national perspective. Receiving standard serum and being able to produce diphtheria antitoxin, for instance, for the Russian Institute, made them a center as their own. We'll just go from this training courses into cooperations and to show how the margins were really very important for the center. So Ehrlich's ties to Eastern European institutions had, due to Scandinavian or US-American bacteriologists, multiple layers, and he had a multiplex networks, a mixture of weak and strong ties that were closely intertwined. Ehrlich's relation to Russian scientists might date back to the 1880s, when Eastern European physicians visited bacteriological institutes at the Imperial Health Office. But at least since he became director of the state-run institute, he might have had further contact to other Eastern European life scientists. However, since 1898, Ehrlich contacted a number of Russian and South European hospital physicians asking if they were treating patients with malaria or other tropical diseases. Like he contacted later on scientists from the Pasteur Institute and from Netley from the Military Institute in Great Britain, if they had patients with tropical diseases. And he offered to send chemical preparations, dyes at that time, to send them, and they would do for him clinical testing of the dyes that were supposed to have a therapeutic impact. The institute itself did not have a hospital ward, so they were dependent on other physicians doing the clinical trials for him. And so this is so obvious. The center, so to say, was dependent on the budget, because without the clinical trial, they couldn't do anything. And later on, it was some sort of a process of verifying the different research results. I will conclude and come back to this photograph and to this institution. What I want to make clear is that networks were not planned strategically. Networks are historical contingent, so to say. And this is, for instance, Salomonsen went to Wrocław. Oops, sorry. I just make a note for me to stop talking. I'm already at the summary. He just came to Wrocław, and they get a closer relationship. They kept contact, so to say. Nobody else. And in 1885, Ehrlich was more or less without a mentor, without a, so to say, tutor. But then everything, so to say, changed. Salomonsen visited him more often. Then he became director of the institute, and then they had a close relationship. Some ties might become stronger for some time, and then they just decrease because they got, as for instance, Torvald Madsen with Paul Ehrlich or with Arrhenius, they have some sort of different scientific differences, and they diminish the contact. Furthermore, some occasional meetings, explanations in the laboratory became, after a number of people asking for explanatory instructions, so that this increase of people making training at the institute becomes even more difficult to keep the contact. And then, so to say, the network implodes because you can't keep their interest. We'll just summarize my talk and going back into the first. Ehrlich was successful because he had multiple layers of relationship which were all intertwined. You have this different sort of relationships. He had personal networks. I just mentioned the pedigrees and the heritage and the kinship of networks. So Madsen was sent to Ehrlich's institute because Salomonsen and Ehrlich were good friends. I mentioned the scientific networks which were important for the knowledge transfer, but also thought collective were necessary within these networks because the communication within the network and the verification of the results and so on and so forth, this is all taking in within a network. And here we have a network of thought collective, of the very new and developing field of immunology, serology, and of bacteriology. And then you have an actor network, which could be best described as stabilization of this process of them evaluation as an actor network. Then you have institutional networks in the sense of you have the Pasteur Institute, you have the Institute for Experimental Therapy and other institutes, but also as you have institutionalized training courses within the institutes. It's not some sort of, yes, you could come and then we look, this is all institutionalized. And you have a mixture of weak and strong ties, so families. This is especially for Ehrlich, so he had a lot of family members, chemists working together. Carl Weigert is a very good example of this. And you have, and this is the margins and the centers, you have the interconnection of centers and margins by and through networks. You could just contact people from Russia, from the United States, from Great Britain. You could correspond with them. And the advantage of their evaluation is you could just send the standard theorem to them, and they could just do the same as you do. And the standard or the theorem evaluation of the German method was more or less thought as independent from the person, from time, and from the place. So that was the idea behind it. But nevertheless, who is the center and who is at the margin depends on the time, the perspective, and the interest. So here you could, as I said, identify probably one of the few people and how they were all interconnected with each other, which is much more difficult if you get into this. Some general remarks. So networks are social and volatile constructions changing over time. And networks as a technology is an own so to say, technology and to build infrastructure. So networks as an infrastructure are vital, as a precondition to transfer information, knowledge, and things. So just to get this in the right way and not coincidentally. And in social constructions, networks are volatile, permanently shifting and mutating. So for instance, as Ehrlich became director of an institute, it was completely changing the whole network. As his mentor died in 1884, this was also completely different. And after the First World War, the whole networks imploded of the German scientists. So this is, so to say, what I mean. And then you have a moral economy of give and take and the reciprocity of networks. So all the people are connected to each other, write letters to each other because they expect something. Because Ehrlich sent an off-print to a scientist asking for, oh, I sent you an off-print, I have worked to this, and he expected to be quoted. Or he expected, he just asked in the next sentence, could you probably send me this or that preparation of I have read your mention in your article and I'm very interested in, and so on and so forth. The specimen, there was a whole economy of sending specimen preparations and so on and so forth. but also letter of recommendations were some sort of a currency. So, and the institutionalization of weak ties and the expansion of the networks by or through training, seminars, internships, and cooperations stabilizes, so to say, also the network as such. And I would say these institutionalized stable networks are a precondition for knowledge transfer and for innovation, so to say, just to spread the idea and to get it approved. Thank you very much for your attention.
Yes. Thank you, Axel. Remarkable phenomenon. Questions and comments. One of the most striking things, perhaps you could comment early on, the center of any given network was obviously the man who had the money. And the money coming from industry, of course, because they were selling, etc. How did that affect the marginalization and the recentralization of new and young people? Was there anything that went on there?
First, before people coming to the institute getting trained, they had to be installed in another institute, so to say. They don't come from nothing. They were always, as I have shown by Metchnikov, they were always recommended, so to say. And they were always already based on their own networks, which get them into the institute.
Unless they were members of the family.
Yeah, exactly. And it costs also money. It was expensive to go there and to pay the fees.
So did commerce pay for most of the research in buying things that were being produced?
That, I would say, depends. Especially at that time, pharmaceutical or the chemical industry became a pharmaceutical company. And they supported Paul Ehrlich and the institute very much. So the Georg Speyerhaus was one of the first German institutions that had been funded by private money, so to say, not by state money. And so that had a large effect. And they were always complaining, look at the United States, look at Rockefeller, look at Osiris in France, and look at the London and Liverpool School of Tropical Medicine. They have so much money and they don't have to do anything for it. They could just do research. So there was always some sort of
Other questions, comments, please. Dave?
I have a similar,
You go
similar question. These institutes, the 50 or 56, were they government institutions? I mean, the pharmaceutical industry was very early days. It was mostly, the chemical dye and the aniline industry. So, and so, following up to Phil's issue, where did the funding come from? These institutes were a government responsibility for the health of the citizens, I presume. And did they then morph into industry, like von Behring, I presume, became an institute, and then Behring-Werke, for the industrialization? And when did that transition happen?
So it's completely different institutions. It depends on the country. For instance, in France and in Germany. In France, you had the Pasteur Institute, but you had in the periphery also other institutions like in Eastern Europe, and mainly it was produced at the medical faculties of universities, or there was some sort of and in Charkov, for instance, it was some sort of a medical academy. So they were more or less semi-private, public, some thought of this. But also William Burroughs and Wellcome, they received, and also Park Davis received standard serum. So it was just who asked for the serum, but there were also regulatory institutions who were responsible for the regulation, just to compare. And for instance, the Jenner Institute, the Jenner Institute was also producing serum. So Dean they had a serum producing department. That was where they started, more or less.
Yeah, I wanted to ask you, obviously, about the methodology, because there are similarities and also differences in our concepts of networks. And I'm not sure if you've experienced that. When we were building our own networks, we were quite puzzled and had a very strong debate about how to document collaboration in a network. So how do you establish a relationship, a tie between two entities, in my case institutions, in your case individuals? And we obviously settled in co-authorship, but we are very aware that that way of documenting collaboration shows some things, but also hide others. I mean, it hides lots of collaboration and relationships that went on around sequencing that are not captured in the publication. So I was quite interested in your use of photographs, you know, to kind of start drawing the networks and identifying the actors. And I just wanted to ask you two things. I mean, how you have combined those photographs with other evidence to decide who was in the network and who was not. And also, what were the role, you have already discussed this a bit, but hearing a bit more, what were the role of non-human actors in the network? Because as you probably know, within science and technology studies, there is a whole debate of whether non-human actors have got agency and can be considered an actor in the network.
Yeah, and probably just to the first part of the question, so it's more or less, the interesting thing is I'm just telling probably a story from about 1900 which has historical worth, one might say. But the interesting thing is my and your story is very, very close and familiar to each other. And I have the advantage of having only a few actors working together, and I don't need all the sophisticated programs just to visualize this, because I have some photographs, and then I can just show how they, with the biographical information, I can just show how they work together. They wrote letters to each other, and this is also, nowadays you have emails and all the stuff, so I have all the correspondence of Paul Ehrlich, for instance, and could just reconstruct the different networks. So this is more or less some sort of a biographical part. Then I have sociological analysis of networks, dense of network strength, and so on and so forth. The number of contacts. But the interesting thing is, Ehrlich had a lot of contact to some people, but there was, I mean, he only sent letters with standard thermo, for instance, and he had some very close relations to other people, and how to differentiate this. And the interesting thing is, of the Pasteur Institute, there are only very little letters, because they were one institute. They were just communicating directly to each other, and you only find literature if Pasteur is outside of Paris and he's just sending letters to Roux, do this or do that, and so on and so forth. So the question is, what can't you see? The interesting thing is also, what don't you see in the network? Who is not involved? For instance, Pasteur Institute, there is no French guy visiting the Staten Serum Institute inauguration, which is very interesting. So this is more or less not only the question of who is in the network, but who is not, and what can't you see. And I think this is one advantage if you're going 100 years back. This is probably a bit more better to demonstrate. The second question is a completely different thing. I have written 2 articles about this actor network and the agency of the different, and how difficult it was. They just started to stabilize everything, and then there was another problem, and they had to start from the very beginning, because there were so many actors, and it was on so many factors, it depended on so many factors to stabilize the system of evaluation. And then they just started in summer, and then they realized the whole system is dependent on temperature, and in winter they had such a lot of problems, and then they restarted from the very beginning. And you can see this in the sidechain theory to explain this practical thing. Because first, the sidechain theory, you had only sidechains, and then you had receptor theory, and then you had this whole menagerie of different aspects and complements and haptophore groups of receptors and so on. So, and this is a very different story. But I would say the story I have told is one part of this overall actor network, because you can just go into the details, into the serum dispensers history, into the pharmaceutical industry. You can go into the toxin and the development of the toxin and the constitution and so on and so forth. So on the one hand, I'm working biographical and a bit sociological, and on the other hand, so this is a clearly A&T thing.
Mm-hmm.
So
yeah, thank you. Very interesting.
Is there a final question before we break? Sir.
Yes, very, very interesting study. I am, at the very least, I learned that next time there's a picture-taking opportunity, I should position myself strategically. I was not aware of what may come
And
later.
labeled yourself number one.
Yes. So the lots of thoughts, but at the core, you have what I retained from your ideas, and a lot of lovely ideas, is this idea of dependency of the center on the margin. In my field, it's very much about how knowledge travels, and we apply network analysis, and there's this idea of a core and a periphery, and the core is where the action is, the large institutional centers. And then there's this assumption that these people talk to each other, and then at the periphery, they ask questions or they rely on the byproducts of the center, and they're very marginal in multiple ways. But what your study demonstrates is that it's a more intimate, multi-layered and intimate relationship, and that the dependency goes both ways, right? And I think that that's really cool. So, and I guess it has come up again for me. It's an eye opener, but listening to Harvey during the break, and like the importance of training is going to be relevant for the next 2 days, right? Because for me, often when something diffuse over network, it's an idea, it's a concept, it's a way of doing things. But these concepts or ways of doing require training, and that is reinforcing of many other ties. And I'm not sure anymore if weak ties is a way to describe it. We have to rethink the concept of ties.
Mhm.
And I think Lawrence talked about it in his preliminary remarks about kind of how these things become important. So thank you. I learned a lot. So I guess it's mainly a comment rather than a question.
If I just, the interesting thing is of the training, it has two aspects. First is you get a training, and the second is you get a personal training. You just realize it. For instance, if I'm going to a conference and I'm seeing a person directly, I just realize, oh wow, there's an article of this person, and I just anticipate it much more than I would have read it. Just, okay, that's an interesting article. So, and this is the same with the building network starts exactly with the training. They know each other. They later on, when they had problems, they just send it to the person they get trained by. And this is, they get a training, but they also get a personal contact. And that's also a very important aspect within the interrelation of the center, so to say, and the margin. Yeah, so thank you very much for your comment.
Thank you very much indeed. I want to thank you all for both your attention, your questions.